So first, before we get into the review, I had basically 3 slides left over from the mitosis meiosis stuff. And basically, I wanted to just talk about one concept around mitosis and meiosis. And this is something called non-disjunction. So basically, it's a definition for you, non-disjunction. And what that means is that you have an improper separation. And so you can see, for example, here's mitosis. And you can see rather than the sister chromatids separating properly, we have, in both cells ending up with 2 chromosomes, we have one cell with 1 chromosome, and one cell with 3 chromosomes. So you end up with a mismatch. So non-disjunction in many cases is actually a lethal event. If you have a cell and it doesn't have crucial genes, the cell dies. Sometimes if you have too many genes, the cell also dies. It's not always lethal 100% of the time, but in many cases it actually is. It's especially if you're an animal. It's tolerated a bit more in plants. Plants don't seem to mind having extra DNA. Sometimes it means they get bigger fruit, or in some cases, this is how we can get like seedless varieties of fruit. And so maybe the fruit itself can't propagate itself, but it can still be eaten. And so there's lots of different ways to do that in plants. Animals tend to be a little bit more sensitive to messing around with their genome. Here's showing meiosis. Meiosis has 2 stages, meiosis 1 and meiosis 2. And you can see, we can either have an improper separation of the homologous chromosomes in meiosis 1, or an improper separation of the sister chromatids in meiosis 2. And so this can also lead to non-viable cells. But there are, like I said, some circumstances where these types of things are tolerated. And there's one condition that you might be familiar with, and it's called trisomy 21. So trisomy 21 is where you have 3 copies of chromosome number 21, so way down over here. And this is tolerated, and the offspring are viable, although these individuals have something that you may know as Down syndrome. And so even though they are, functional animals, and it's a non-lethal mutation, they do have various physical and mental defects in terms of development. So there are other conditions like this. This is just kind of one of the better studied ones. So that's it for mitosis and meiosis, other than for the midterm. So what I want to do is talk about the midterm today, and go through these review slides and review a few concepts as we go along. So midterm is Wednesday, and if you are doing some studying, I am available. You can see this note here. I am available tomorrow afternoon and Wednesday morning. I'm relatively free, so if you want to go over anything, just send me an email, and we can set up a Zoom link. See someone has a question?
Yeah, I just have a question about like the sample questions and the sample like lockdown browser. I completed like the sample questions, but it doesn't show me which ones I got right and wrong.
Oh, okay. I thought I had that. I thought I had that configured to tell you which was right and which is wrong. So I'll have to go into the settings later.
Okay.
If I don't remember, let's say by tomorrow, please remind me tomorrow morning if I haven't done it by then. So let me just write a note. Thought I had checked that off, but I guess not. Sorry about that. So midterm 2 review, like I said, if you have any questions, please get a hold of me. I have a little bit of time between now and Wednesday if you want to go over any questions. So I showed you this slide before. This is the plan for the midterm. And so I have some sample questions along these lines. It's pretty similar to midterm 1, with the big exception being we are doing this mitosis meiosis question that we were talking about last day. So if we have time at the end here, we'll do another mitosis meiosis question. So just a reminder, we're doing the lockdown browser. I do have a test question there that you can try to make sure it works. I know sometimes people update their computers or get a new one or whatever. So I would encourage you to give it a try before Wednesday, just to make sure things are working. I think most people, it went fine. But we did have at least one person with issues last time. All right, let's talk about the midterm. So 6 topics, and I kind of have a slide or 2 in each of these, and I'm going to spend a bit more time on the last 3 topics, so 10, 11, and 12. So let's get into these right away here. So topic 7 was on the cytoskeleton. So make sure you know what each of these 3 things are: microtubules, microfilaments, and intermediate filaments. And I want you to know examples of what those things do, right? So microtubules, for example, are part of, they make up the eukaryotic flagellum. So these are the types of things to know. So not a huge lecture, just lots of kind of, funny words and things like that. But like I said, make sure you do know examples of what each of these things do. And also review the flagella structures. I'm definitely going to have some questions asking you about these things. I can't recall, but I think last semester, or maybe it was the winter semester, I had a long answer question comparing and contrasting these things. So just to give you an idea of what you're looking at. Here's a sample multiple choice question. It says intermediate filaments are A, assembled from actin monomers. Nope, that would be microfilaments. B, play a major role in the contraction of skeletal muscle. No, that is also microfilaments. And this is actin and myosin. Are permanent cell structures? Yes. And that would be the nuclear lamina is what we were talking about. Those are the structures that are holding the nuclear membrane in shape. Guide the movement of organelles. No, that would be microtubules. Are the largest of the cytoskeletal structures. That would also be microtubules. So the correct answer is C for that one. Yeah, it looks like I have it in bold. So topic 8 was on cellular order and energetics. So here's a little cartoon for you. You can see the guy, he says he has a bottle of beer and is next to a mug of coffee. And he says the beer is getting warmer and the coffee is getting colder. And he's not so happy about that. And he says, someday the opposite will happen. And on that day, I shall mock the laws of thermodynamics. And his friend says, not today. So unfortunately, yes, we do have thermodynamics to think about. So topic 8 was a whole bunch of kind of related concepts around energy and definitions. So some things to know are catabolic, anabolic, endergonic, endogonic. There's also this delta G thing to know a little bit about that. So for example, you can see this delta G is positive, and so that means that reaction requires energy. So we're coupling that with the hydrolysis of ATP, and that's what's shown here. ATP is being added, and it's basically a 2-step reaction. This is kind of similar to what I drew on the whiteboard. They're kind of showing the steps a little bit differently, but is that we are the net is that the overall reaction is negative, which means it's losing energy and it is favorable. So remember what this is called. This is called energy coupling. And the other word we used was cellular work. So lots of words to know what all these things are, just in general. Okay. So sample multiple choice question, it says during an endergonic reaction. So end means in. So this means the energy is going in. Oh, there's the answer. Meant to talk about these a little bit. So energy is going in. So no, this means energy is going out. This means energy is going in. Entropy means disorder. So no, order is decreased, energy is released. So endergonic means energy is going into a system, or energy is required for that system to progress. So like I said, make sure you go through that, that topic and just spend some time on the words and definitions on what those things mean. Topic 9 was on enzymes, and same thing as topic 8. I want you to go through that topic and just kind of make sure you know what some of the definitions are. Some of the definitions, for example, we talked about things like substrates. We talked about things like cofactors. We talked about inhibitors. So lots of things to consider and most of it's relatively straightforward, especially if you've talked about enzymes a lot in like high school. One other thing that we talked about was regulation of phosphofructokinase. I'm not expecting you to know a lot of details about it, but know what it is. It's found in glycolysis, and it's a key regulatory enzyme. That's kind of mostly what I want you to know about it, meaning that it's turned on when we want more glycolysis, and it's turned off when we want to turn down glycolysis. Sample question: define active site. So what is an active site? So remember when I told you with the short answer kind of written questions, I'm looking for more than just one-word answers. Most of these questions, I'm marking them on a scale, meaning you can get it sort of half right, 3/4 right. For this particular one, you got to tell me something about an enzyme, and that's where the enzyme binds the substrate, and it's also where catalysis occurs, right? So there's kind of two parts to that. So cellular respiration, topic 10. I want to review the reactions of this. I'm not going to review all the parts of the mitochondria, but it would be a good idea for you to do so. And you can see how them all labeled there. Hopefully now it's pretty straightforward. So like I said about this, how much detail to know. I really want you to focus on kind of the net of what is going on in each reaction. Sort of, what is feeding into it and what is coming out, and why these things are doing so, right? So the things, like I said, to focus on are carbon, electrons, and ATP. So let's just talk about that as we go through these. And like I said, we're going to, on the midterm, there is going to be a question, and it's going to ask you, it's going to show you a pathway, and you have to identify it. So if you can review the reactions in such a way that you can identify them, and then it's going to ask you some questions about it. So I'll kind of get into some details now on this. So here are the pathways of cellular respiration. And so we had 4 pathways here to learn a little bit about. And of course, they're all making and consuming different things. So let's talk about glycolysis for starters here. So glycolysis, like I said, if we're following all the things that we are using, it basically starts off with glucose and finishes off with 2-pyruvate. Okay, and remember this is the carbon, this is a 6-carbon compound, and this is a 3-carbon compound. And so the other things I told you to pay attention to are the ATP. So we're going to overall, we're going to make 2 ATP. And to make 2 ATP, we need 2 ADP and 2 inorganic phosphate. So I'm going to stick those in and make 2 ATP overall. And then the other thing I told you to pay attention to are the electron carriers. So the electron carriers here are is NAD plus. NAD plus will pick up electrons and a hydrogen, and it becomes NADH. So those are the 3 things to pay attention to, right? So on the exam, I could ask you questions about any of these things here, right? I could ask you, what is NAD plus for? So your answer is that NAD plus is going to pick up electrons during the oxidation of glucose. It's going to get reduced, and it's eventually going to go to the electron transport chain. What is ATP for? So ATP is produced, and what is it going to do? ATP is going to be used for cellular work. Cellular work. What is the pyruvate used for? Well, the pyruvate is going to move on to the next step, and it's going to be oxidized further and liberate more energy and more ATP in the long run. So like I said, be prepared to answer anything about this. I could ask you, what is ADP for? And you would say ADP is the ingredient of ATP. We need it to make ATP. You could also say it's a nucleotide as well. So there's lots I could ask you just from this bare information here. So pyruvate oxidation, as I mentioned, is the next step. So pyruvate oxidation, cram this in here. Didn't leave myself a ton of space, but we start off with pyruvate. So remember, pyruvate has 3 carbons. So I'll put that down here, and we are going to go over here to acetyl-CoA. Acetyl-CoA. So acetyl-CoA is actually, we've only got 2 carbons left at this point. So what does that mean? It means we're losing a carbon somewhere, and we are losing that carbon as CO2. So again, where does the CO, what's the point of the CO2? The point of the CO2 at this point is it's a waste product. And as animals, we're gonna send that out and eventually expel it out of our lungs. So what else is going on here? No ATP is being made. I don't need to do that part, but we are going to oxidize pyruvate and reduce NAD+ and make an NADH. So once again, the NADH is going to go to the electron transport chain. And that is pyruvate oxidation. So kind of 2 things going on. We've got the NADH being made, and we've got the CO2 being released. And I guess there's one thing that we're forgetting is our coenzyme A. So coenzyme A is going to go into this thing. So hopefully that makes sense. There are different ways to look at these reactions, and that's sort of how I look. You can see I have all the reactants on the left and all the products on the right, and I'm just doing arrows to show that. Okay, citric acid cycle. So there's the last step here. So the citric acid cycle, we have acetyl-CoA going in. Acetyl-CoA. And okay, I'm going to go in there. There's my cycle. There we go, my arrows. And try to squeeze everything in here. So coenzyme A is released. And we're going to release some carbon dioxide. So there's the carbon part. We're going to make some ATP. So we actually make 2 ATP. I guess I didn't leave myself very much space. To make 2 ATP, we need to put in 2 ADP and 2 inorganic phosphate. Sorry about that. I didn't mean to cram that in so tightly. We're going to make, what is the net here? Sorry, it's 1 ATP. It's 2 for 2 acetyl-CoA. Sorry about that. The net is 6 NADH. 6 NAD plus becomes 6 NADH. So there's my arrows going in and coming out. And then we've got FAD becomes FADH2. Okay, so like I said, I'm not so concerned about the exact stoichiometry and how many numbers here. It is useful to kind of keep track of everything. But in the end, right, we've got some carbon here. This is our carbon right here. We've got some ATP being made. And then we've got some electrons that are eventually going to go to those electron, to the electron transport chains. Okay, so move on here a little bit. There's our oxidative phosphorylation. So I'm going to draw a little bit of this as well. Actually, I think I have this on the next slide. So why don't I just, where's my slide? Sorry, I had a slide. Let me see if I can find it for you. Hold on a second. I know I had it here somewhere. Must have deleted it by accident. Okay, well, I'll just go back to here and I'll have to draw something for you. Not a problem. Okay, oxidative phosphorylation. So I'm not going to draw the membrane. I'm not going to draw anything fancy here, but let's think about what's going on here. We've got some NADH going in. Okay, like I said, like before, I'm going to do the reactants over here. We have FADH2, and we have oxygen, and we have ADP, and inorganic phosphate. So the NADH is going to basically get oxidized to NAD+. FADH2 is going to get oxidized to FAD. Those electrons are eventually going to end up on water, and we're going to get H2O. So if you want, you can throw in some protons here as well. Like I said, I'm not balancing the formulas. And in the end, we are making ATP and a whole bunch of it. So that's my kind of oxidative phosphorylation in a nutshell. This, of course, takes place around the the inner membrane of the mitochondria, and you've got that, you've got that proton mode of force being produced, and you've got that whole electron transport chain, which is the oxidative part, and the phosphorylation is, of course, the ATP synthesis part. Okay, so I'm not expecting you to know, like I said, all the steps here. There's a whole bunch of steps and different complexes and all that, but this is what I want you to focus on, is what is basically going on here. We're oxidizing these electron carriers. The electrons ultimately end up on oxygen, forming water, and that makes the proton mode of force. The proton mode of force is used to drive ATP synthesis. And that, of course, is where most of the ATP is being produced. So just a reminder, know these pathways, know what's going on, okay? Just let me know if you have any clarifications around or what details that you need to know. So one other pathway, or I guess two other pathways that we talked about, is what if there is no oxygen? And so if we have oxygen, of course, we can go to the right, and you can see over here, we've got all these pathways we talked about, where we're going to do pyruvate oxidation and then the citric acid cycle, and eventually make a whole load of ATP in the mitochondria. If you have no oxygen, let's say it's in human muscles or in some microorganism, we might do something called fermentation. So fermentation is over here, and this happens in the cytoplasm, and it can produce many different types of products, kind of just depending on what organism you are. And the two that we talked about, of course, were producing ethanol and lactate. So I think I have the pathway here. Let me see here. Maybe I don't have the pathway there. So if you remember from lab 6, we did ethanol fermentation. So the products in that case are ethanol and carbon dioxide. And if you're an animal, we don't do that. We make lactate instead. So this is the kind of thing that you could see in like these short answer questions, right? And so as I mentioned, these short answer questions, they could range from something like this, or they could be fill in the blank, or give 2 examples of, or tell me what happens during the stages of prophase, or something like that. Or sometimes they're asking questions like this. And I feel it's important for me to have the short answer questions in there because it really does reward the people that have worked hard and studied really hard. But sometimes they trip people up, and I find this one here I've had on tests before, and people think it's a complicated question, right? Turns out it's not. Go back to that previous slide for a second here, right? What's going on here? There's no oxygen. So that's half of the answer here. If you say something about there being no oxygen, I'm going to give you half a mark for this, right? This is the full technical correct answer. But depending on exactly how you're trying to answer this question, you don't have to have all this stuff, right? So like I said, half a mark for this no matter what. This part's non-negotiable. There's not enough oxygen in where? The muscle cells, right? So chemically, this is what's going on. Pyruvate is used in electron acceptor to regenerate NAD+. Okay. You could always say something like, so there's not enough oxygen, and therefore pyruvate is not fully oxidized or broken down. And I might give you a full mark for that. I'd have to think about it. That's generally what's happening, is you're looking at a partial breakdown of pyruvate rather than a full breakdown to carbon dioxide and water. Like I said, there's different, not necessarily all one right answer on this, but usually part of it's non-negotiable. You have to have this thing about not being enough oxygen. Okay, so I'm going to move on. Here's a sample long answer. So as I mentioned, I've given this one out before. Because we're doing everything online, I'm kind of avoiding most diagrams. Although you will have to make a diagram for me for this test for the mitosis meiosis question. So make sure you do have some extra paper so that you can do that question. But here's an example of a long answer question asking you to compare the prokaryotic and eukaryotic flagella. And so remember, it's just kind of actually very similar to what we saw in the last lab, right? So describing the structures, describing the motion, does it rotate, does it flap back and forth? And, where does it get its energy from? Those kind of things, right? And so, do a good job, you get 5 marks. So remember these 5-mark questions, right? You should have at least 5 things, at least. Kind of depends on what the things are. If you're just saying something small or something big, I'm not going to give you a mark for that. The more details, the better, obviously. So let's talk about photosynthesis. I know photosynthesis is a bit more daunting than respiration. And so we're not like, we didn't go into it as much detail, but there are some things to know about it. So overall, there's the reaction of photosynthesis, right? So we are starting off with carbon dioxide and water. Of course, we also need energy in the form of light. And we are going to produce some sort of carbohydrate. The big one to remember is G3P, which is glyceraldehyde 3-phosphate. Oxygen is, of course, a byproduct. I usually ignore water because it consumes more water than it produces. And I'm not really so concerned about water. I'm concerned about energy, and I'm concerned about carbohydrates and ATP and things like that. Water is not as important. Not that it's not essential, but it's just less essential to the understanding of this whole thing. So there are the major steps of photosynthesis. So if you need to know one thing about photosynthesis, this is the slide to memorize. And you can memorize it in the 2 halves, the light reactions on the left and the Calvin cycle on the right. So I was going to just kind of rehash the light reactions here in a very brief way. I think in the lectures we had like 7 or 8 steps. I'm looking at this in just a few steps, right? So our light reactions, so first thing to remember is what these things are. These things here are called the thylakoids. And the chlorophyll is in the thylakoid membranes, and that's the actual green part of the plant. That's where the chlorophyll is actually kept. So remember, we have light goes in and light splits water. Okay, so maybe what I'll do, actually just cross that off for a second. What I'll do is the same thing I was doing before. I'll have the left side as the reactants and the right side as the products. Okay, so reactants, we've got light and we've got water. And the water, of course, gets split into oxygen and it gets split into electrons and hydrogens, but I'm not going to show the electrons freely because the electrons are going to join our electron carrier, which is NADP+. So that gets made into NADH. Some of the electrons also end up in ATP. So of course that would include ATP and inorganic phosphate. And there's our ATP. So that's really the light reactions. You can see that's kind of mirroring what's going over there on the left. Chlorophyll is an intermediate, meaning that it's not consumed. It's regenerated. Even though you're stripping electrons off chlorophyll, it does get regenerated. So same thing like I was saying before, I could ask you about any of these things, right? So, what is NAD+, NADP+? It's an electron carrier. What is this should be NADPH. Sorry about that. And what is NADPH? That is an electron carrier, and that is going to go, where is it going to go? This time it's not going to an electron transport chain. This is not respiration. This is going to go to the Calvin cycle. So what about oxygen? It is going to be basically a byproduct here. I guess you could say it's going to maybe someday be used for respiration, but we don't know that. But for the sake of photosynthesis, we are done with the oxygen. What is the water used for? It is the source of electrons. So, we can talk about all these things. So make sure you're able to converse and talk about what's going on in here in terms of photosynthesis. So there are the light dependent reactions, a little bit of a different picture from what I showed you in class. The class one I showed you was from the textbook. This is from the Khan Academy. So I'm not sure if everyone is familiar with the Khan Academy. Khan Academy. They have their own website, and they're also on YouTube, and they are basically educational videos from pretty much every topic you can possibly imagine that you would cover, at least an introductory level sciences and many areas of humanities and whatnot. So if you want to, if you're reviewing biology or any of your other classes, go check out Khan Academy. They've got lots of good stuff and very well put together educational type videos. If you go to their website, I think they have quizzes and stuff you can do. It's been a while since I've checked them out, but they made this nice picture, and you can see all those ingredients that we were talking about. Here's the light, here's the water producing oxygen, and our final products are NADPH and ATP. And the final thing to remember is that this happens, of course, on the thylakoid membrane. So what about the Calvin cycle? So reactants we can put over here, products over here. Reactants, we've got carbon dioxide, and we've got the products of the light reaction. So ATP, NADPH, and that's pretty much it. So let's make these things into something. So we're going to make G3P. That's our carbohydrates. ATP is going to get broken down into ADP and inorganic phosphate. And NADPH is going to be broken down into NADP+. And that's kind of the Calvin cycle. One thing to remember is that enzyme. The enzyme's name is Rubisco. And this all happens in stroma. So the stroma is this part of the chloroplast, kind of the area outside the thylakoids, but inside the inner membrane. It's kind of like the cytoplasm of the chloroplast. So maybe one other thing to remember is that the Calvin cycle is sometimes also called carbon fixation. So that's a pretty common name to be called for the Calvin cycle. Okay, so I have a big question I want to go over with you. Not this one, but another one here in a moment. And one that I told you I was saving till the review session here. But here's a question kind of getting us to think about some of these words, right? And if you haven't studied, this is going to be a hard question. If you have, maybe it's not so bad. First word there is chemiosmosis. What does that mean? That's just a fancy term for talking about the proton motive force, basically, and ATP synthesis. So if you know that, then you know this is good, this is good, this is good, this is good, and this is not good. So make sure you study and you'll get these questions. So there's the answer, Rubisco. So sample true and false. The purpose of the light-dependent reactions of photosynthesis is to produce ATP and oxygen. So this is false. Oxygen is not the purpose of the light-dependent reactions. Purpose is to make ATP and NADPH. Oxygen is the byproduct, right? So make sure you read the questions carefully. Okay, so this is the question that I wanted to cover in today's review. And usually this is kind of something I do in the board. So instead, I'm going to do it in a Word document. So I've started making a Word document here. And I'm going to make a table. And you can see the question is, by the way, if I were to give something like this on a midterm, I wouldn't leave it so open-ended. I would probably say, discuss the following, right? But anyway, let's talk about this because I want to talk about respiration and photosynthesis a little bit and compare a number of things. So maybe the first place to start is this, right? So where are mitochondria and chloroplasts found? Because this is something I find people mess up on. I think I had this on a test last year and people said animals and plants. Well, you know what, animals, yes, they have mitochondria, right? But so do plants. Okay, it's not that animals have mitochondria and plants have chloroplasts. Mitochondria are found in animals and plants and protists. Okay, oh, and fungi. Okay, so they're found in eukaryotes, right? Chloroplasts are found in photosynthetic organisms. And that would include plants, but don't forget all of the other ones as well. Don't forget, we have algae and we have various protists as well, can also do photosynthesis, okay? You're probably thinking, okay, what about bacteria? Some bacteria do respiration and some do photosynthesis. They don't have chloroplasts and they don't have mitochondria. They do the processes and they usually use their plasma membrane to do so. Sometimes their plasma membrane has a kind of a folded in surface to do so. So that's just a side note for you. It wasn't in any of my notes. Okay, so second thing is, so what happens here, I guess is the question. So what happens in the mitochondria? We have respiration. So which processes in particular? We have pyruvate oxidation. We have the citric acid cycle. And we have oxidative phosphorylation. And each of these citric acid cycle happens in the matrix. Oxidative phosphorylation happens in the inner membrane. So one other thing that we did mention is something called beta oxidation. Also happens in the mitochondria. So that is breakdown of fats. I mentioned that at least once earlier in the semester. So maybe I'll throw that down there. What happens in the chloroplast? We got photosynthesis. And that's two reactions. We got light reactions and the Calvin cycle. And in many cases, we have also storage of starch and other things. Starch storage. Okay, so I know the question is asking about electron transport chain and ATP synthesis, but we'll get there. Don't worry. Okay, so let's talk about electron transport chains. Okay, so if we're going to talk about electron transport chains, we want to talk about electrons. So where are the energy-rich electrons from? Okay, so let's think about this for a minute. In mitochondria, we are oxidizing food. So remember what oxidizing means. Oxidizing means we are losing electrons. So our food is losing electrons. So that's where the food comes from. So we can say that our food is from organic compounds. And maybe I'll put in brackets that we eat. Right, so such as glucose. Remember, electrons are not just free things. They live in mostly chemical bonds. What I'm going to do actually is maybe I'll come back to photosynthesis in a minute. And I'm just going to throw in another one here. So where do the electrons go is the next question. Where do the electrons go? And the term I use in lecture for this is final electron acceptor. So where do they go? They come off of glucose, okay? They end up on those electron carriers, so they end up on NADH and FADH2. So maybe that's the next question. What are the electron carriers? But I'll get to there in a moment. And so where did they go after that? They go through the electron transport chain, they zip through the electron transport chain, and they end up on oxygen. Oxygen is the final electron acceptor. So remember, here was our formula. We had O2 plus 2H plus, and I'm not going to subscript everything. This is just going to be my, I'm just going to type it like that, and we get H2O, right? There we go. So oxygen is the final electron acceptor, and oxygen gets reduced and becomes water. So like I said, and the next one is going to be, what are the electron carriers? Okay, so in respiration, we have NAD plus, and it gets converted to NADH. And we also have FAD, and that gets converted to FADH2. So let's go back to photosynthesis for a moment. Maybe this is the easy one to answer for photosynthesis. What are the electron carriers? So one called NADP+. So it's very similar to NAD+, except for it has an extra phosphate group, by the way. That's basically what it is, NADPH. Okay, so now let's go back to photosynthesis, and it says, where are the energy-rich electrons from? Okay, so we got to go way back to the beginning of photosynthesis. Think about our light reactions. We have light, okay? A light does not have electrons. It hits chlorophyll, and chlorophyll donates electrons to electron transport chain, but the chlorophyll gets its electrons from water. So water is the electron source. So H2O, and so remember water, it's H2O, and it gets split. That's the term we use. It gets split into 2H plus oxygen plus 2 electrons. So sorry, I'm not subscripting it and superscripting everything for you there, but hopefully that makes sense. We've been through it in lecture already. So we have a gap in our table, and maybe you see where this is going, right? If respiration makes water and consumes oxygen, photosynthesis produces oxygen and consumes water. So where are the electrons going in photosynthesis? They are ending up in bonds in organic compounds. So chemical bonds in organic compounds. Okay, so not quite done here, because we still have a couple things to talk about ATP synthesis and the electron transport chain. But like I said, I'm just trying to get you to think about these things in a slightly different way, because I think it's important to the deep understanding of what's really going on here. Okay, so the next question is, where is the proton motive force in each of these? So another way to ask that question is, where are the protons pumped to? And so in the case of the mitochondria, we are talking about the intermembrane space. So that's the place between the two membranes. In the chloroplast, this goes to the thylakoid space. That's where the protons go. I just need to learn how to type today, apparently. So that's inside the thylakoids. And 2 more quick questions. Where do the protons flow to? So basically meaning, where do they go after the intermembrane space? And in that case, in the mitochondria, it's going to go back to the matrix. So back to the matrix, and maybe I'll put through ATP synthase. And what about in the chloroplast? There's no real place to go. They can't go any further in, so they got to go out. And so this goes back to the stroma and also through ATP synthase. Okay, I have one more thing here on this table. And the last question is, what is the ATP used for? So what is the ATP used for in respiration? We make ATP and then what? Well, it's used for cellular work. Remember what that means? It means we can transport things, we can move things, we can do energy coupling. What is ATP used for in photosynthesis? It is used for the Calvin cycle. And what does the Calvin cycle do? Calvin cycle uses it for carbon fixation, meaning it takes carbon dioxide out of the atmosphere and puts that carbon dioxide into organic compounds. Okay, so there's some things for you to think about when you're reviewing photosynthesis and respiration in terms of what is going on here. You can see there's some parallels. There's some things that kind of, it looks like one is doing the opposite of the other and all that. And this is why we kind of learn these things together, because there's a lot of parallels in these particular reactions. May never see photosynthesis again, which is too bad because it's very interesting. But like I said, that's the reason why I don't go into a ton of detail in photosynthesis. Most of us may never see it again unless you take a botany class or something like that, a plant cell biology class or something. It's also possible you might see photosynthesis if you take some microbiology classes, because there are a lot of microorganisms that do interesting things with photosynthesis. All right, so a few minutes left, just enough time to talk about the cell cycle. And hopefully we'll have enough time to do one of these sample problems here. So there's our PowerPoint. And topic 12 was the cell cycle. So we covered both mitosis and meiosis. So make sure you review what is going on in each of the steps, because there will definitely be worth a few marks on the test, whether multiple choice or short answer. I'm going to skip this because I think I want to do a problem first. And I have, you can always go back and take a look at that if you like. I want to take a look at this problem and make sure that everyone knows kind of what they're doing here. So like I said, I'm going to have one of these problems. It's going to be with 5 marks. Sometimes I might ask you something else about the stages. Like I might ask you, what is happening during prophase, or, in terms of make sure you know your terminology, sister chromatids versus homologous chromosomes and things like that. So this one says we have a diploid cell. It undergoes meiosis to produce gametes containing 2 chromosomes each. So the first part of this is asking us to draw the parent cell at late interphase. So I want you to basically tell me how many chromosomes we have, right? So read the question. It says we are producing gametes. So a gamete is a sex cell. So it's a haploid cell. So that means N is 2. So that means that 2N, the diploid cell, is 4. So in this case here, I'm just looking for 4 chromosomes, right? And like I said, it's more or less just counting, showing that you have things correct. Remember that number 4. We're going to start off with 4. In mitosis, we're going to have 4 chromosomes, and we're going to get 4 chromosomes. And that means in meiosis, we're going to start off with 4 chromosomes and end up with half of that. We're going to end up with 2. So let's do this problem here. So we'll do mitosis first. So what was the number? 4. All the chromosomes, I'm going to replicate them, right? And they're going to have a sister chromatid. So they're all going to look like that. So what that would be 1 chromosome, or with both sister chromatids still attached. So I'm going to do that 4 times, 4 metaphase. So I'm just going to draw them as stick figures like this: 1, 2, 3, and 4. And I'm going to see if I can change my pen color here. There we go. And I'm going to draw my spindle fibers. There we go. There's my spindle fibers. So what's happening during anaphase? During anaphase, the chromosomes are separating. So I'm going to do that. I'm just going to change it back to the red. I'm going to draw 4 chromosomes: 1, 2, 3, 4. And the sister chromatids there are separating. Draw the spindle fibers. And the spindle fibers are going to the end to where the centrosomes are going to be. And then after that, you just basically show what's going to be in each cell. So let's see here, pointer options. It's not easy to change the ink color for some reason in this. Go through a couple of submenus. So 1, 2, 3, 4, 1, 2, 3, 4. Okay, so that's mitosis. Let's do meiosis. So remember, meiosis is going to start off with 4 chromosomes. The difference is that they're going to be arranged differently during each of the stages. So in this case here, we've got, here's my metaphase 1, and you've got these homologous chromosomes here. So they're going to be like this. I'm just trying to make them fatter so I don't have to change colors for the spindle fibers. So there we go. I'll just do it like that. So there's 1, 2, 3, 4. Now, oops, they're just, I don't know how I erased that, but Okay, so they should be touching at metaphase, so I'm just going to stretch them out a little bit here. They're still touching. There's my spindle fibers, going to each end, and there we go. So the ones on the left are going to go to the left, the ones on the right are going to go to the right, and I'm skipping a bunch of phases, but now I'm in a metaphase 2. But remember, at this point, since things are dividing, now we have 2 cells, we're going to have half the amount of DNA at this point. So here is first cell with 2 chromosomes now, and we still have the sister chromatids together. It's going to look something like that. And there's my spindle fibers going in there. And then lastly, my final gametic cells. Remember, I'm going to have 2 chromosomes in each of them. So there we go. So that's what the mitosis meiosis question is going to look like. So hopefully that's not going to be too difficult. If you know what you're doing, if you can count, and sometimes people double things or half things at the wrong point, and that's where it gets a little bit weird. So do take a look at these problems. Maybe I would recommend maybe go and do this one here on your own, and then you can look back later and see if you got the same answer that I did. And if you can do that, you're in good shape. So hopefully everyone is going to be in good shape for Wednesday's test. I know some people are kind of scared by this one, and the material is a little bit more difficult. But I'm hoping you're all better students now. You know, you've been through one of my midterms. You know, hopefully how to study and what to expect. And so you just need to do the work between now and Wednesday if you haven't done a bit already. So it looks like we're out of time. So thanks for coming out. And like I said, get a hold of me if you have any questions about the material before Wednesday, and I'll be happy to help.